Hi everyone, Fitz here. In today's video, we'll be going over your acid-fast stain. The acid-fast stain is a differential technique that we use to identify specific bacteria. The bacteria of interest are going to be Mycobacterium, TB, tuberculosis, as well as AFB, which stands for acid-fast bacilli. Now, there are a few different types of acid-fast staining techniques. The one that we're going to be going over today is called the ZN method, in which we use heat to target the waxy layer of these bacteria called mycolic acid. And when using this technique, that waxy layer, mycolic acid, has a high affinity for the primary stain and has a repelling effect when it comes to the decolorizing step. So the first step that you want to do here is prepare your water bath. And the way that this works is you will have a beaker of water with a screen on top, and then you will place your slide with the bacteria on top. You will have some sort of apparatus underneath here, usually heated by a Bunsen burner. As that's heating up, you will want to heat fix your bacteria. Once you've heat fixed the bacteria onto your slide, you will want to cover it with bibulous paper and cover the bibulous paper with the bacteria underneath with carbon fusion. Carbon fusion is going to act as your primary stain. With the presence of the heat, what will happen is that waxy layer will melt a little bit and will then be able to take up the primary stain. So as you're heating this up, you will want to steam it for 5 minutes, allowing the carbon fusion to be taken up by the mycolic acid. And if the bibulous paper ever runs dry, you just add a few more drops of carbon fusion. Never let this run dry. It can go anywhere from soaking wet to damp, but you never want it to dry out. As soon as that 5 minutes is up, go ahead and turn off the heat, dispose of the bibulous paper, and wash the slide out. Next, you'll want to use acid alcohol. This will act as your decolorizer. And a good question to know the answer to on test day is, why don't we use the same alcohol as we did in our gram stain? As soon as the appropriate time has lapsed, go ahead and wash off the acid alcohol and add methylene blue, which is going to be your secondary stain. As soon as you've let the methylene blue saturate the slide for the appropriate amount of time, wash it off with water and blot dry. Then grab your microscope and put your slide underneath for further observation. Now, when you have your results underneath the microscope, you're going to see one of two things. You are either going to see the color pink or the color blue. Unless your lab professor like mine pulls a mean trick on you and gives you a mixture of bacteria, you will see pink and blue. Note that the pink color means that you have an acid-fast positive bacteria because it took up the carbon fusion and was resistant to the decolorizing step. If you see blue, you're going to note that your bacteria was acid-fast negative here because it took up the secondary stain. So the color coordination is opposite to that of the gram stain, in which blue was the primary stain and red was the secondary stain. And here are some questions that you can expect on test day. Now feel free to answer those questions down in the comment section below. If you found this video helpful, please leave a like and subscribe. And don't forget to check the description for additional study materials, and feel free to ask any additional questions down in the comment section below. And I'll see all of you in the next one.